Okay, good afternoon. I'm George Froudakis, the chairman of the chemistry department of the University of Crete, and I would like to welcome all of you, and especially our students, in this very special colloquium. Our honored guest is Professor Jean-Marie Lehn, who was awarded in 1987 the Nobel Prize in Chemistry and is one of the most influential people, not only in organic chemistry, but in chemistry in general, since his work has been addressed in many different fields. We are also really glad, as he belongs to our greater family of the chemistry department, since he was being awarded a honorary doctorate in 1989, 2 years after his Nobel Prize. So we welcome Professor Lehn once again in our department, and we are really grateful to him as he immediately accepted our invitation. Unfortunately, our rector, Professor George Kondakis, cannot participate since he is busy with an emergency in the university, but he has asked me to welcome you and thank you for accepting this invitation and give us this seminar that we are all of us waiting to hear. I would like now to give the floor to Professor Dinos Neochoritis, who had prepared a few introductory slides before we proceed to Professor Lehn's talk.
Hello also from me. Professor Lehn, I'm Dinos Neochoritis, I'm an assistant professor here. We had a contact those days. And I also would like to mention that it's a great honor for us, and especially in these difficult times due to pandemic, that you are here and you will inspire the next generation of chemists or the young people. So let me share my screen. So, Professor Jean-Marie Lehn was born September 30th, 1939. Apparently, we have the same birthday. I was also born on September 30th.
Oh, really?
Yeah. In Rosheim, where Rosheim is, it's a city, it's a small medieval city, 25 kilometers southwest of Strasbourg. And it's very nice. It has very nice wines, as I read a little bit. It's a wine-making town. So this is France, Else is somewhere there. And Rosheim is a small city right there. And in 1950 to 1997, there was the first big dilemma, I would say, as I'm reading in your page in the Nobel laureates webpage, chemistry or philosophy. And Professor Lehn was very good in philosophy, ancient Greek, Latin. So luckily for us, Professor Guy Ourisson persuaded him to go for organic chemistry. And luckily, he was dragged by the beauty of organic chemistry. In 1957 to 1960, he went to the University of Louis Pasteur in Strasbourg. He did his bachelor. And then in 1960 to 1963, he obtained his PhD degree under the guidance again of Professor Guy Ourisson. His first paper was published in 1961 about conformational and physical-chemical properties of triterpenes, an NMR study. Then he moved to Harvard University for a postdoc. He worked with Robert Woodward in this memorial synthesis of B12 vitamin. He worked with another legend of organic chemistry, Hofmann. He did his first computations, the known Woodman-Hofmann rules. And then he went back, 1960 to 1970, in Strasbourg. He was appointed as an assistant professor and ranked till and raised to 3 ranks and went full professor in 1970. Then in 1971, the lab was moved to the Lebel Institute. And in 1979, he was elected to the chair of Chimie d'interaction moléculaire. Forgive me about my French, but it's a long time since I had it, at the Collège de France in Paris. And of course, the big moment, everyone knows, Nobel Prize, Chemistry, 1987, along with Donald Crum and Charles Petersen, for the development and use of molecules with structural and specific interactions of high selectivity. So let me say a few words about the new people here. Charles Peterson is known, he discovered Crown ethers. This is the known polycyclic ethers where they can bind small or bigger, depending on the size of the Crown ether, metallic ions like lithium, sodium, potassium, and even bigger. Jean-Marie Lehn discovered analogous compounds. He called them cryptans, I suppose from the Greek word, right? And then they looked something like that. They are polydentate compounds, and the important thing is that they can capture certain types of molecules, bigger or smaller or bigger, depending again on the size of the cryptant. The important thing is that they create chemical entities with great impact, both in biology, medicine, and materials. This is one of the most known cryptants, where you can see that one small ion of potassium is captured there. But the important thing, to my opinion, is that Professor Lehn really showed that chemical reactions back then often occur through the influence of molecules that have cavities and pockets, where there other atoms and molecules can be joined, and then become something bigger. I was lucky enough to have in my master's degree in Thessaloniki a course about supramolecular chemistry. And let me tell you that supramolecular chemistry is the new area of Professor Lehn that introduced back then. So I really remember a quote from a famous architect back then, Louis Kahn, that even a common, ordinary brick wants to become something more than it is. And that's true. Brick want to become this nice architecture.
Oh, very nice.
So Professor Lehn let us know and proved that even molecules want to become something bigger. And a lot of nice supramolecular self-organized structures have been created. Highly functionalized molecules that today it is proved more than ever that they have a tremendous impact. So the supramolecular self-organization that was introduced first time today is applied everywhere. Polymer chemistry, nanoscience, nanotechnology, design and properties of programmed supramolecular systems. And to my opinion, the Nobel Prize in 2016 about molecular machines is an extension of this. One other important aspect, also to my opinion, is the dynamic combinatorial area that Professor Lehn was also introduced, and I was lucky enough to be a part of this while in my stays in Netherlands under Professor Hirsch. So there, initial building blocks create something, a random library of interchanging species, and then our target, which most of the times is a receptor or a protein, selects through, again, self-organization, the best target. And this indeed has found a profound application in drug discovery. So a lot of things, helicates, grid-type entities, multi-compartment nanocylinders, everything. Professor Lehn was and still is very active. So 60 years of research, more than 300 collaborators from over 20 countries, 1,000 publications and review papers. This is a picture from a recent group meeting that they had, probably institute meeting. Professor Lehn is not unknown to the chemistry department, as the chairman also said. He was awarded a honorary doctorate 2 years after his Nobel Prize. Professors from here went to his lab as visiting professors, and proudly we have also in our curriculum a supramolecular chemistry course. So I don't want to delay further. We have a small photo surprise for you, and with the help of Professor Orfanopoulos, we'll go through them.
Good afternoon. Thank you, Dinos. Good afternoon to all. Dear Professor Jean-Marie Lehn, we are happy to have you again here with us, even through a computer screen. You visited us 31.5 years ago. So to refresh your memory, as well as to many of my younger colleagues, I prepared a very short PowerPoint with photos from that time. You gave 2 lectures actually in 1989, The first one was for the inauguration ceremony for the honorary professor of our department, and a second one for the students. This is the second one here in the slide. And it was the time we were working with overhead projectors and blackboards. Next, after this, we had the celebration dinner at a restaurant across the Knossos Palace. And next, the acting chairman at that time, Professor Karabatsos, on behalf of the department, he offered you a gift, which was actually the disc of Festos. Next.
I have it. I still have it.
Okay, good. I wanted to ask you. So next, the first actually rector of the university, Professor Dimitris Markis, made a toast to you. Next. And of course, your best wishes and some comments on our department. The next day, we decided to make an excursion in South Crete. We visited first Festos Palace. As you see here, most probably Professor Karabatsos is listening of what you read about this place. Next, we visited the famous Asian caves at Matala. That was a place that hippies were visiting early 60s. In the middle is my good colleague, Thanasis Koucholelos. He looked very like a teenager in this picture. And actually, let me make a comment here. Many of today's faculty members that time were either in high school, some of them in elementary school, even in 2 or 3 of them, even in kindergarten. So the last slide is actually a dinner in a fish tavern in Matala Bay. And that was the last picture I wanted to show. If you noticed, one of us is missing every time from the slide because was taking actually the picture. So please visit us again in person. We left many places to visit from this beautiful island that we stay here. So I don't want to take any more of your time. I will pass now the podium to our distinguished speaker. The title of his lecture is Steps Toward Life, Chemistry. Professor Lehn, please take the podium.
Thank you for this presentation, this introduction. I was very interested, and I think if you can send me the PowerPoint, the two PowerPoints, that would be very nice, because there are pictures here which I don't have, or at least I didn't find them. So it would be very nice, and I have very nice memories of Crete, of course. It's a pity that I cannot be there today. And in the meantime, I have been several times in Greece because I have a number of co-workers who have been working here in Strasbourg, who are now have positions in Greece, and I have seen 2 on the screen. I will mention them a bit later again. And so thanks for the invitation. This possibility, of course, I immediately accepted because I think Greece was always one of my favorite places where science sort of started. So let me just get into the game. What you have told about what we have done, of course, for the students, they are not aware of that. So my talk here, and you have asked me to do that, is a general public lecture for students who even can be people who are not in chemistry, in other fields. And so let me start with the title, which I've said, Steps Towards Life, Chemistry. This means that I will try to show you that chemistry plays a most important role in all these aspects of life. But of course, we don't know yet how life has occurred. But in order to get to the point, we will start very, very early, so early that it was right after the Big Bang. Right, the Big Bang was 13.7 billion years ago. It was a big explosion. Our universe started. At that time, the universe was extremely hot. There was no chemistry. There was energy, which gave then particles formed. After 1 second, there were already particles, as you can see. And then the universe cooled down and the particles formed atoms. This was the age of physics, no chemistry. The basic physical laws controlled the beginning of our universe. And then when the atoms got together, they formed molecules, constructions based on atoms. That's about here where chemistry started on our planet. And this is the age of chemistry. Then these molecules, by different processes, became bigger and bigger, attaching together and forming larger molecules, forming also compartments. And within these sort of compartments, which were around, a new property emerged, which we call life, about here, and that's the age of biology. Now, it wasn't finished because the biological organisms evolved, became more and more complex until they reach on our planet this organism which has a property which is thinking. For me, this is even a more important property than life. Of course, you cannot have thinking without life, but the fact that we are here talking together, that's because we have a brain and we think. And that is the steps towards life, and I could even say towards life and thought. Now, this is not a slide which is dark. This is intentional, because I would like to remind you that our universe is 68% dark energy and 27% dark matter, which means 95% darkness. Not very funny, but that's not our problem, because there is 5% visible matter. And this is, as I like to say, the matter that matters. We are part of that. We are part of the 5% visible matter in our universe. The rest, we don't know. Physicists, cosmologists begin to know more and more about, but don't worry, we are part of the 5%. And in fact, this 5% visible matter is, as I say, the matter that matters, because it makes also us what we are. Now, if we want to go back to this evolution of the more and more complex forms of the visible matter, of course, we start with divided matter, then the matter becomes condensed, then it gets organized, living, thinking, and maybe something more complex even than thinking. Now, I would like immediately to point out also that on my previous slide, like here, the end of the screen is not the end of evolution. That means that we are a point in space and in time, and the universe will continue to evolve. So there are people around the world now who say, oh, we have to stop everything. We can't continue like that. This is a very short-term view. The universe is 13 billion years old, and the universe doesn't worry about us. But we can have a fantastic influence. And we come back to that because we can think about the universe. So this is the evolution towards more and more complex forms of matter, maybe even more complex forms, more complex as they go with time. And maybe this question marks indicates that someday there may be a more complex way of behaving, of thinking, of whatever it is, another property, which we cannot know about right now because it's beyond our thinking. Now, the big question then we have to ask is that how does matter become complex? How is it possible for matter in the universe to become more and more complex? It means that our universe is constructed in such a way that matter does become complex. It's a tautology. Because we exist, that proves that matter can become complex. And it's a property of our universe. So how does one go from an elementary particle to a thinking organism? Are there perhaps even higher forms of complex matter? How can you address such question? You can address it by inventing what mankind on our planet, this very, very small piece of dust in the enormous universe. Mankind has developed science. And physics deals with the laws of the universe. Biology deals with the rules of life. These are not laws, these are rules, because the laws are the physics. And all the rules of life depend on the laws, of course, of the basic laws. So what is chemistry doing? In fact, we can claim as chemists that we are not just making molecules, materials, drugs, and things like that, which is very nice, of course, very nice to be able to do all that. But that is not the most important. Basically, what the mission of chemistry is to do this, to build the bridge from the general laws of physics to the expression of these laws in things like life on the planet Earth. And this is, of course, a fantastic work. We have to do studies and a goal, a challenge to try to understand how we can bridge this, how we can express this in specific organisms and specific matters, specific behaviors, psychology, and all this is linked to it. So I have only cited here, of course, three parts of science, but they are all linked together. But these are, I would say, these are the three sort of two pillars and the bridge which has to be constructed between the pillars of the general laws and the expressions of those laws on our planet, but also on other planets. And I'm convinced there is life on other planets. There are so many billions of planets around, and there's life very, very probably somewhere. So what is the answer to that question? In fact, there is an answer which we can all understand, although we don't know exactly how it happened. It happened, at least that is what I think, by self-organization. I mean by that, it doesn't tell you how it happened, but it tells you, you can understand what it means. It tells you that it happened by itself on the basis of the structure, the laws of our universe. Our universe has a structure such that it will self-organize somewhere. And this is, okay, that's a claim, but that is my conviction, that our universe has a self-organization driving power which makes it more and more organized in certain regions. And one can even claim, make another step to say that it's a cosmic imperative, that our universe is built in such a way that life is unavoidable, not an accident. It just happens to be part of the game, part of the general plan of the universe. So this self-organization I mentioned, for the moment we have still to look at it in two ways, because the physicists have not yet provided us with a general unifying theory. So on one hand, there is the self-organization on the large structure of cosmics, of the cosmic self-organization through the gravitational forces, Einstein forces, which operated on initial inhomogeneities in density and rates of expansion at very early times after the Big Bang. this is not my field. So what I tell you is what I know from cosmologists. So this is the structure of our universe, which results from the action of gravitational forces. But this is not our talk, the matter today, because this is part of what the cosmologists study, physicists study, black holes and stuff like that, and the gravitational waves and all that. What we are interested in is complex matter, which is molecular matter, which results from the action of electromagnetic forces, which then structure the building blocks of our universe in a random structural combinations, and then under the laws of the laws of thermodynamics, these organize, and some are somewhat better, meaning having better organizational properties. Of course, we don't know exactly what that means, but it means that it has evolved in such a way that it gets more and more organized and more complex. And this is what we sort of are interested in when we think about how life could evolve and how thinking could evolve. So this chemical evolution of matter, of course, started much before biological evolution. We all know about Darwinian evolution, but there must be, and there has certainly been, a chemical evolution of non-living matter. Because you cannot, it's very difficult to imagine that you jump from protons to a living organism. There are steps of increasing complexity until you reach the threshold of life. And then something happens, which we don't understand yet, but we will understand at some stage, and life appears. This possibility of reproduction, of carrying information from one generation to another one, and having a metabolism. Now, wait a minute. This chemical evolution, when life appeared, became what we call the Darwinian evolution. But Darwinian evolution itself is also chemical evolution because, as you know, and we see later, that this Darwinian evolution is an evolution in a chemical entity, which is our genome, which is chemistry. We come back to that. So chemistry at this stage in my talk, we can define as the science of the structure and transformation of matter, living matter as well as non-living matter. We will see a bit later that there's another property which is very, very important. Now we start very early because mankind had people thinking about what was going to happen. So we had the Greek philosophers, the Greek thinkers. You may have noted, I didn't point it out, that on my first slide, there was this statue sitting at the end of this arrow on steps towards life. That was the thinker of Auguste Rodin, a French sculptor. And of course, what is well known is that in your country, 2,500 years ago, there is a set of people who were thinking about what was around us. What does all that mean? We can call those philosophers. I have a tendency to call them thinkers. Philosophers is a certain way of approaching things. Thinkers is using your brain, your thought, to try to solve problems. And let's have a look. 2,500 years ago, there was this person, Empedocles, who proposed that that matter was made of 4 elements: fire, air, water, earth. You know all that because these I also use for talks in other countries, in China, in India. You know that, of course, you have learned it from the start. But just to remind you of it, and these elements can then combine to give properties. So that's already not so bad. It's an attempt to explain. But there was another person who went much further. This is, of course, Democritus. I have a great admiration for Democritus because, as you know, he sort of proposed that matter was a particulate. It was, and which could not be cut into smaller pieces. And you know, atoms. Now, the story is, and maybe in the audience there are people who know more than I know about that, But the story is that Demokritos has had this idea from something like an experiment. And maybe I don't know exactly what it was, but let me just tell you that if you go around the street and want to convince somebody who doesn't know anything about chemistry, who doesn't know what matter is made of, and want to convince that person that matter is particles, you can do the following experiment. You take a glass of water, you put in a piece of sugar. What happens? The sugar disappears, it dissolves. If matter were continuous, it could not do that. It means that matter can mix together, and so there must be particles which mix. For me, this is a way of convincing people that matter is made of particles. And it is said that Demokritos got this idea from this type of observations. I don't know if it's true or not. I have read quite a few things, but one has now no writings of Demokritos. Now, I wouldn't like to forget, of course, who is the person who is called the Prince of Philosophers, Aristotle, and who had a fantastic influence on the thinking of the Western world, of course. For me, Demokritos had, of course, a very important insight. Aristotle was, in some respects, broader, but I think he had a very strong influence on the world. Demokritos came up again when atoms were really discovered. Let me just, I'm sorry, I'm a bit long, it will take longer than an hour, I guess, but I would just like to point out that if indeed Demokritos has based his hypothesis of the particulate nature of matter on an observation, then it is fantastic. Because you can have philosophical theories, but if you base them on an observation, that is a whole different goal, a different study, and a different ball game. Now, we talk about Demokritos, he's talked about atoms, and over many years, then the so-called scientists or whatever they were, people who were trying to understand what was going on around us, they discovered that there were bricks of matter indeed, which later on were related to atoms, of course. And these bricks formed a sort of a collection which didn't seem to present any order until the middle of the 19th century, which I would say the middle of the 19th century to the beginning of the 20th century was really the age of science, chemical science, because at that time, chemists were thinking about what they were handling, and it was very difficult to understand what this matter was made of. In the middle of the 19th century, suddenly order came. Mendeleev, there were others too, but he went by far further than the others. He proposed, and he had good reason to propose that all these bricks which had been discovered can be organized, can be put in a table. This was in 1869, and I want to show you his first paper on that. Here it is. This paper is one of the most important papers in science everywhere, anytime. This paper indicates that on the basis of, in English, the relationships between the properties and the atomic weights of these bricks called the elements, one can organize them. And this is the first table of this organization of the elements, which he published in a Zeitschrift für Chemie in 1869. And you see, you can recognize, as chemists, you can recognize these things which are here. You have here boron, carbon, nitrogen, oxygen, fluorine. You can go and then as a row, fluorine, chlorine, bromine, iodine. And there are even question marks, because Mendeleev was so convinced that he was right that he said there are things lacking. I put a question mark. And of course, these were found later. So I insist again, because I want to do that, I really like to do that. This paper is one of the most important papers in science ever. And this is now called the periodic table of elements. Now let's elaborate a little bit on that. Let's think about this. Nowadays, that is what we have. The bricks of visible matter. I insist on visible because this is this 5% of the total matter of the universe. This is now called the periodic table of the elements. Now, for me, the astonishing thing when I look at this is that this table is complete and shows you what any combination of the elements, any living organism, any matter, anywhere is made of the same things. Why can one be so sure about it? It bothers me because, you know, science cannot be dogmatic, cannot say that's what it is, because we know science evolves. We know that science is corrected from time to time. It's evolving, it's getting better and better. But here, one cannot escape the fact that the table is complete. And why is it? Just because the sequence of numbers is complete. 1, 2, 3, 4, 5. There's nothing between 1 and 2, between 2 and 3, 3 and 4. And what is it here? 1 proton, 1 electron, 2 protons, 2 electrons, and so on, 3, 3, and so on, so on, so on, until uranium. Of course, the other ones are artificial elements, and we don't talk about those. And there are no living organisms probably made with those things. But what we can say is that the table which you see here is represents the bricks of any material object, including any living matter in the universe. A carbon atom is a carbon atom anywhere from here to the end of the universe. And we know that also because when people look at galaxies, look at and so on, they can find optical indications that certain elements are present. And I would also like to insist on one thing. A carbon-oxygen bond has the same strength everywhere. It has not another strength on another planet. It has the same strength. And this is absolutely incredible that we are sitting here. I'm sitting in front of my screen and you of your screens, and we can say that we know what visible matter in our universe is made of. Every time I say that, and I repeat it many, many, many times, I'm astonished. You're really taken back, saying this is incredible to know what everything is made of. And that is thanks to science. And this is also for the chemist something important because it's the playground of chemistry. Chemists are like children. They play Lego, but they have a very, very, very complex and interesting Lego set, the elements. And with these elements, they can do an infinite number of combinations, obeying, of course, the laws of physics. You cannot do anything. There are laws which control what you can do and what is stable, what you can fabricate in given conditions. But that's what chemists can do. And it's a fantastic thing to be a chemist because you can play with all these things. This is in your hands. You take a bottle, another bottle, and you can play with it. So this is the composition. What are elements? The composition of matter. Then you have to transform matter from one entity into another one. And the first person who proposed the rule was Lavoisier, when he said, rien ne se perd, rien ne se crée, tout se transforme. Nothing is lost, nothing is created, everything is transformed. Of course, if you think about the transuranian elements, scientists have been able to create things which did not exist. But this is, of course, with respect to the elements which are the stable ones of the periodic table. So this is a basic rule without which there is no chemistry, of course. But this chemistry which we have here, then we have now the composition of matter, we have the transformation of matter, then along the way, and I haven't time to talk about all that, people have noticed, scientists, so to call, if you call them this way, have noticed that the elements can bind together for forming certain combinations. And these combinations then were considered to form the basis of the visible matter. But it is not enough to consider that these elements are linked together in a certain way. They also have a shape. And this was proposed in 1874 by two chemists, Van 't Hoff and Lebel, independently, Van 't Hoff in the Netherlands and Lebel in France, when they said that chemical objects, molecules, are not just a graphical connection between elements, but they are an architecture. In one of the presentations at the beginning, there was a talk about Louis Kahn's architectures. That's exactly what chemistry is also. It's an architecture. It has a shape, and this is the start of stereochemistry. Of course, talking about life, we cannot miss mentioning Pasteur, because he was the first one to understand one of the basic properties of most molecules in a living system, which is the fact that molecules can have a relationship between each other, like a left hand and a right hand. And here again, Greek comes in, molecular chirality, kairos, as you know, is hand. And so this is, of course, very important thing. Pasteur did many other things. And in fact, he's very important right now because we look about vaccines, yeah? But okay, that's another story. So this is sort of very, very brief walk through some historical points in chemistry. There are many, many others, but of course, we could spend days and days and days talking about. So this path from the atom to the molecule led to the buildup of molecular chemistry. And molecular chemistry is, as you know, but it's also for the students, as I said, formed by the connection of atoms through strong bonds, covalent bonds, called covalent bonds. Now let's look at some milestones in this molecular chemistry. In 1828, Friedrich Wöhler in Germany made in the laboratory the molecule called urea. And he made it from ammonium cyanate. Urea is present in living organisms. Ammonium cyanate is not in living matter. So by doing that, he did two very important things.
Oh, okay.
But do you want it?
Amazing.
Do you want it on mute?
Please switch off your
I
microphone.
didn't recognize that. But
Okay? Is that fine?
Yes, go ahead, Professor Lehn.
Okay. That's strange. Should work. So he made urea in the laboratory from ammonium cyanate. And in this way, as I said, he did two important things. First of all, he did a chemical transformation, ammonium cyanate into urea. And another thing, he showed that you can make a molecule present in a living organism, urea, in urine, from another chemical entity which has nothing to do with a living organism. In doing so, he showed that there is no magic separation, no vital force, as it was thought at that time, which prevents from making molecules from living organisms. He showed that all of that is chemistry, living or non-living matter. Then 100 and many years later, chemists were able to build up this very complicated molecule compared to urea, vitamin B12. This resulted from the combined efforts of two groups in the world, one at Harvard University, directed by Robert Burns Woodward in the US, and the other one, Albert Eschenmoser at the ETH in Zurich. This was considered at that time as the top, the Himalaya, the Everest of chemistry. Of course, nowadays, molecular chemistry has evolved further, and one wouldn't make this molecule probably the same way anymore. At that time, it took about many, many years. I mean, many men and women years. It took 10 years, but every time 10 or 20 people, so it is something like 100 or people who have participated in this story. And I worked on this. That is my part in this synthesis, which I made. I was postdoc with Woodward in 1963-64. This is not to show that it has to brag upon that to show that that is it, what I made. No, it's just to show the students that when you do science, you bring a stone to an enormous construction, which is construction of knowledge. And here it's just a construction of a molecule, but if you are a scientist, you bring a stone to knowledge, a small one or a big one, but it is your stone and you contribute to the work, the studies, the goal of humanity to try to understand what we are doing around, what we are doing here, and what matter is we are made of. Now, as I said, this molecular chemistry is continuing, is still very much alive, and many new reactions, new transformations, new molecules, new materials have been made, new procedures have been invented in the last 50 years or so. And this will continue. But we can ask a question then, is there another aspect we should look at? And I would like to introduce that in the following way. I show you here a cancer cell, the blue sphere, and two killer cells, the purple units here. The killer cells have a goal to find out which cells have become cancerous, have become transformed, and when they find it, they have to destroy it. And of course, they should not make a mistake. If they make a mistake, you have a big problem. They might either not kill a cancer cell, or kill a healthy cell. This is no good. So how do the killer cells know that the other one is a cancer cell? Of course, as you see, they touch each other. That means that the membranes which define these entities, and which are made of molecules, like a soap bubble, let's say, they also contain characteristic molecules, proteins, which gives some notion about the identity of the other partner. Another example is here, a white blood cell, which is surrounded by blue particles. That's particles of the HIV virus. When the HIV virus hits a white blood cell, it can infect. And so what has happened? What, sort of say, if I may say so, tells the particle, the virus particle, that the other guy is the goal and can infect. Something must happen between these large bodies made all of molecules. And what happens here must be between the molecules, not inside, between the cells, between the virus and the cell. And this is touching, and this touching brings molecules in contact. So something happens between molecules. And this then led us to think that beyond the chemistry of covalent bonds, the molecular chemistry, there is another chemistry, a supramolecular chemistry, which is a chemistry beyond the molecule, which relies on contacts between molecules through interactions, which are not the interactions making up molecules, but which make up larger constructs which rely on these non-covalent interactions. And three main properties then have been studied over this last 50 years or so, now a bit more, which are, on one hand, first of all, the most basic one is molecular recognition. We come back to that. Reactivity, how molecules can react with one another, and transport, the way they can be transported through barriers, like a cell membrane or an artificial membrane in some artificial vesicle or some other type of entity. So let's have a look at what is this most important property at the start, molecular recognition. Sorry, excuse me. So molecular recognition requires, of course, first of all, an interaction between the molecules. They have to bind. If they don't have an interaction, they ignore each other. They don't know there's a partner. So there has been to be a physical interaction first. But that is not enough. A very important property, basic property, is information. This process, which happens between molecules, is a process of information, because you cannot have a specific interaction. You cannot have a recognition between molecules if there's not an interaction, information present. And this can be represented in very simple way. That's the simplest way. Of course, it's much more complicated, but basically, it's a sort of a complementarity in geometry and in the interactions between the objects which partner together. And this was already explained, proposed a long time ago by a very strong and nice image provided by Emil Fischer in 1894. Emil Fischer proposed in a paper in 1894 that for an enzyme to act on its substrate, they have to fit together like Schloss und Schlüssel. This was a sort of a mechanical image, of course, because it was the age of mechanics, if I may say so, in this end of the 19th century. But it is true, it has to fit. And of course, we now know that things are adaptable somewhat to some extent, but basically, it is a strong image, which is true. And when I give public lectures, I usually stress that if you go home and remember that molecules, when they get together, they play games of locks and keys. You have already learned something very important. Let me just point out that Emil Fischer was a PhD student in Strasbourg, got his PhD in 1874 under Adolphe von Baeyer. You know, Europe has changed a lot. At that time, Alsace, my region in France, was part of the German Empire because the French had lost the German-Prussian War, and so that part of France was integrated into the German Reich. But that's all right. I mean, we had these people like Fischer, we had Adolphe von Baeyer, we had many other people. Even Röntgen was a professor in physics here. He didn't invent the X-rays at that time in Strasbourg, that later in Germany, but he was around too. So it was a very, very, very scientifically, very advanced and very active place. And nowadays, of course, all this has disappeared. We are all Europeans, aren't we? And this is the most important thing. I like to bring that also to the young students who may listen. We are Europeans. If we don't stick together as Europeans, we won't have any influence anymore. Influence is through sticking together as a united Europe. Now, molecular recognition. Let me just mention what is the most important process of recognition, and at the same time, an extremely simple one. The double helix of DNA is carrying what makes a living organism, the genetic program which defines what the organisms will be. It makes a difference between a tomato and an elephant, or a dog and a human being. And how is this transferred? How is that information stored? Very, very simply. It's a long strand shown here for chemists, that's obvious. And on this long strand, sort of a polymer of rather simple groups, not very complicated, phosphate, and here is sort of a small sugar, are attached letters, just 4 letters, which chemists have given names. This one is adenine, this one is guanine, thymine, and cytosine. I guess all of you know that, of course, but I want to insist on the fact that these letters are very simple. And these letters are enough to carry the information by the sequence in which they are arranged. So 4 letters are enough to define the genome, the identity of living organisms on our planet. There may be other planets where there are different kinds of letters, different ways of doing this, but this is the way it happens on our planet. Now, since we are talking about steps towards life, let's look at one of these letters, adenine. Adenine is a pentamer, is a combination of 5 simple molecules of HCN, hydrocyanic acid. And you can see here HCN, HCN. Of course, there are some rearrangement, but there are 5 carbons, 5 nitrogens. And of course, you find HCN in the interplanetary interstellar space. And so I can easily imagine that this is just chemistry going on around in the universe. It had a long time to do all that. If you add some water, you get guanine. In some other transformation, you get then cytosine and uracil. So we see that this is rather simple. I don't want to talk about proteins today, it's too long. But then you have to read this information. If you store it, you have also to read it, to process it. And this happens again in a very simple way, just by touching between two partners. Adenine can undergo a double interaction represented by these 3 points here with thymine or uracil. Guanine undergoes a 3-pronged interaction with cytosine. So it's touching by 2 or touching by 3 interactions, which is a binary system which makes it possible for the information present in the genome to be read out and processed. 2, 3, 2, 3, like a computer functions with 0, 1, 0, 1. Current, no current, current, no current going through. Here is 2, 3, 2, 3. Now you may say, okay, this is very trivial. How come? How come? Just because probably evolution looked for something which will be robust and simple. It is quite possible that evolution has tried many other things. Maybe, I don't know, maybe 6, 7, 8 letters, many ways of combining, but it probably was too chaotic, wasn't robust enough. And so the simplest is just 4 and 2 by 2 pairings, and that is enough. So if I want to summarize at this stage, I have to say that the storage of that chemistry is also an information science. The information is stored into the molecules, and the processing of that information occurs at the supramolecular level by touching between molecules. And this is, of course, the basic notion that chemistry is the science of informed matter. Matter has information, and chemistry has to understand how to store and how to process and everything that results from this information processing. Now, let me just give you a very, very quick indication why we started all this. And it had to do also, it was mentioned that I was very much interested in philosophy and so on. So the interest in philosophy was linked immediately, well, a little bit later, on the fact that, of course, thinking is in the brain. So is it interesting, what can a chemist do about the neural system? But of course, in the 1960s, middle 60s, this was a bit complicated. So I was looking for some way of getting into, let's say, neurochemistry. So the initial motivations were to try to do something, you have to participate in this whole game. And of course, a simple, a very important one, but rather simple feature of the nervous system is the propagation of the nerve influx. Why was this attractive? Because it was simple enough to be accessible by chemistry. Why? Because the propagation of this nerve influx rests on the change of concentration of sodium and potassium ions across nerve membranes. They go in and out, and the propagation of this signal along the nerve makes the action potential, which propagates the signal from one end of the nerve, from the head of the nerve to the terminus. And I found that, of course, sodium, potassium, that's accessible to a chemist. Of course, there are molecules doing that. That was much more complicated, but at least we can say, all right, there must be molecules which are able to make the difference between a sodium ion and a potassium ion. Let's try to understand how this is possible. It's difficult to understand it at that stage, how biological molecules do it, but can we understand the principle, the basic principles of how this can be done? And this then raised the question of how can one selectively bind and transport sodium and potassium cations through a membrane. in this case, the membrane of the axon. Now, I cannot insist about all the things which had been done at that time, but it was obviously, if we sort of think about what Emil Fischer would have done if he had been around at that time, would have said, okay, sodium, potassium are the keys. Let's see what the lock can be to recognize these keys. Now, this means that one would like to build a molecular receptor, a molecular entity which has a certain cavity, which is the lock, bind one of the keys here, one of these keys. And then the one which would fit among these three is, of course, the red one. And we can call this the supramolecular entity then. The combination of the right key with the correct lock is a molecular recognition process, and many studies have been conducted, and we come back to that, in numerous laboratories on what is molecular recognition, how can one understand it, and how can one expand on it. So let's look back at these very simple cases. Sodium, potassium, this was of course the simplest one could imagine. The alkaline metal cations are one of these columns of the periodic table. There is just a sphere, a ball, with one positive charge from lithium, sodium, potassium, rubidium to cesium of increasing charge. So how can one recognize a sphere in the collection of these spheres? And especially these two, sodium, potassium, which are so important in living organisms and in the nerve system, and in addition, which are close to one another. So it must be very precise recognition, otherwise you make mistakes. And this raises a problem. How can one selectively bind and transport spherical ions? How can one perform spherical molecular recognition? We then designed what was mentioned in the introduction. PowerPoint was playing with locks and keys. Then if the keys are the spherical entities, then we have to make the locks which corresponds to that. And here is these are the scriptans. This is, note that it's Greek again, because I did 4 years of Greek at high school, so I thought I have to base my things on Greek. And Greek doesn't change anymore. It's not like when you call it in French, in German, or in English. This is something which remains. This is around for many, many, many years. So cryptans are molecules containing a cavity, a crypt, and we can tailor these cavities so that they get small, they are small, larger, and even larger. And in fact, they fit then lithium, sodium, and potassium. And that's indeed the case, and this has been shown over the years. And maybe I just can indicate that this was the work of two of my co-workers at this time in the end of the 60s, Bernard Dietrich and Jean-Pierre Sauvage. And Jean-Pierre Sauvage was part of the 3 people who got the Nobel Prize in 2016 on molecular machines, which was not a bad thing. I was very happy about that. So chemistry, then we can look at it, and I know, so of course I cannot tell you everything about supramolecular chemistry, it would be again days and days. Molecular chemistry was starting, we can take as an initial milestone the synthesis of urea from ammonium cyanide by Böhler, 1828, making molecules and making specific molecules which are able to recognize a substrate. And then in 1978, I proposed the name supramolecular chemistry in 2 papers because it appeared to me that we are on a level of 1 step beyond molecular chemistry, talking about what molecules do when they get together, when they associate, when they talk together, when they mix together and do something with one another. This then is a binding of a substrate through non-covalent interactions, leading to supramolecular entities which perform this very basic process of molecular recognition, of transformation of one molecule by another one, or of translocation or transport, like what happens between cell membranes, across cell membranes. And this then allows you to think about devising functional systems on the supramolecular level beyond the molecule, And this field has, of course, then expanded very, very much over the years. Many of my collaborators, I had a number of collaborators in Greece who have worked on this type, certain aspects of these problems, and many, many laboratories around the world have and are working on it, and it will continue. It looks like molecular chemistry is still very active and will continue. Supramolecular chemistry is now going ahead and will, of course, continue and be sort of a classic for the years. Now let's talk a little bit about applications, because a new area, okay, fine, but what is it useful for? The first thing is, of course, it's good for knowledge, but maybe we can also apply it. Basic research gives you knowledge. Applied research helps you, makes you apply that knowledge. The first very important feature is that supramolecular chemistry is a basic contribution, the basic feature which is important for drug discovery. What is a drug? A drug is a molecular key for a biological lock. So the recognition process between the key and the lock is very, very important for drug discovery. It's basic. Without that, you cannot do any drug discovery. And of course, understanding how this happens is basic. So there is no life without molecular recognition. Every molecule, as you're sitting in your room, you know that, of course, but it's always good to say it and to repeat it. The molecules, they know what to do, when to do it, where to do it. And without that, there is just no life. Then we have also worked for a number of years on trying to develop many other of those entities. I just give you some results from our work, but of course, many, many other applications have emerged. We made also sort of cryptans and cryptates with the europium ions, which then, when they are in this other type of cage, they can absorb UV light and emit red light. They can be used as a label for medical diagnostics. And here you see immunoanalysis system, which is now used in a number of hospitals. Important also is gene transfer. Gene transfer is a transport process, as I mentioned at the beginning. Translocation, transport is a basic feature of supramolecular chemistry, picking up a unit and letting it through a membrane. And here in gene transfer, we had studied quite a lot transport systems in artificial, just trying to understand what happens. But then came up also this very important application, so to say, of understanding how to bind an entity and help it to go through a membrane, which is gene transfer. And this resides in making synthetic vectors binding to a gene, a DNA, a piece of DNA, and helping it to go into a cell so that it can go to the nucleus and then be expressed as the RNA, and then finally lead to the synthesis in the ribosomes of the protein corresponding to the gene. I like to insist on gene transfer for a reason, which is, first of all, it's quite clear that it is potentially a way for correcting genes which are defective, like, for instance, in cystic fibrosis, which is affecting children, in cystic fibrosis, where a chloride channel doesn't function in the lungs, and then by gene therapy, one can try to correct for that. There are other applications. I would also like to insist on its use in biotechnology. And this comes to that hot topic, which is called genetically modified organisms. Let me express very strongly that we need genetically modified organisms. We now know how to do it, and people are afraid of it. There's no reason to be afraid. We know how to handle that. We need genetically modified organisms. We will use them. And this sort of perturbations which now occur by some people saying, oh, we don't want that because it's touching life and so on. Life, we know what life is. We know how we operate. We know what genes are. We can do that. And we will do. I'm sure we will do it because it will help us enormously. Okay, we can discuss that more if you want. What about biomaterials? There are also supramolecular applications in biomaterials, especially supramolecular polymers, which are can be made biocompatible and biodegradable. Let me just give you an example of what has been done on that level. First of all, let me give an example about drug discovery. And here I like to very quickly say something about hypoxia and normoxia. The fact that one has, the fact that one in certain regions of our body, there can be a lack of oxygen. And we were looking for a way, for a drug which would restore that oxygenation, because it would be interesting for cardiovascular disease, for cancers, because tumors are usually, not all of them, but usually in hypoxic state and lacking oxygen. So a compound which would restore normal oxygenation would be of great interest in cardiovascular diseases and cancers. And here we have then developed this compound, and I like to see, I saw them in the audience. I like to here point out Konstantina Filaktakidou and Alex Kumbis. They were around, I saw them in the pictures. They are now both professors at the Aristoteles University in Thessaloniki. And I would like to thank them for the work when they were around, because this compound has the ability of increasing oxygen delivery by red blood cells to hypoxic tissues, putting them back into normal oxygenation conditions, and also to act, this is more complicated, but also to act on signaling pathways which have a great importance in the generation of tumors by inhibiting a certain kinase and so on. So, but let me just give you an example, because this compound is now in phase 2 in the clinic in the University Hospital in Zurich. And but some work, the initial work before it went to patients, to the clinic, the work was done in Strasbourg. And let me just show you work done in Strasbourg on liver cancer, hepatocellular carcinoma. Here is the control rats which were implanted with a liver cancer tumor. And so you see, after 1 week, 10 rats were implanted. These are the control set. 1 week, 3 weeks, 5 weeks, enormous tumor formed, and all the rats were dead. I was between 7 and 11 weeks. Then you do the same experiment, but you use what is commonly used as a chemotherapeutic agent, doxorubicin, which usually people are given. And you see here that, okay, it's a little bit slower. It's slower, it has some effect. But then 7 weeks, 11 weeks, and then it goes, in fact, usually once the organism, so the tumor is adapted to the presence of doxorubicin, then it really explodes more or less. It develops very fast. 11 weeks, all the rats dead. And then what happens with ITPP? As you can see, you implant the tumor, and after 1 week, you treat the animal by injection into the peritoneum with ITPP solution. 3 weeks, 5 weeks, 7 weeks, 15 weeks, no tumor anymore. This was dramatic change and is an eradication of implanted tumors in the liver. Now, before we get sort of hyper excited and so on, we were, of course, but we have to realize that these are implanted tumors, that they are more sensitive to whatever happens around them than tumors which are generated by spontaneous evolution in an organism. And of course, things are not so simple as it would seem here. When you see that, you say, okay, the question is solved. That is not the case. It is much more complicated. Medicine, biology, medicine are much more complicated because we are all different individuals, and implanted tumors are not spontaneously formed tumors. But they offer a very good hope and another access. And as I said, this compound is now in phase 2 clinical trial in Zurich. Now, what about biomaterials? We developed also in 1990, we proposed to develop a category of polymers which were called supramolecular polymers. This was published in 1990. I say that because you will see there's something interesting about the date. The first paper was in 1990. It came from my group when I was professor at the Collège de France. They did the work there. Then it happened that 23 years later, in 2013, I got an email from a small startup company called Xeltis, which had developed this supramolecular polymers, made them biocompatible. So these supramolecular polymers were then used as materials for cardiovascular implants for the surgical treatment, that means the reparation, the reconstruction of the heart of children who had a severe congenital cardiac malformation. And this was done indeed. And now I show you the little girl who was the first to receive it. Here is Dominika, at that time 4 years old. She was implanted by Professor Leo Bokeria at the Bakulev Scientific Center for Cardiovascular Surgery in Moscow on October 23rd, 2013. 23 years later, after having proposed and introduced this notion and the first compounds of supramolecular polymers. Okay, you would say that took a long time. Yes, it took a long time. But you know, when you want to introduce something into a human body, you have to be extremely careful. So many, many controls, many steps to go around, to go through, a lot of financing to be found, but eventually it happened. And of course, this was a fantastic satisfaction to be able to see their children running around with supramolecular polymers in their heart, sitting here. Now she's now older, she's now 11 years old, and she's fine as far as I know. They're both very happy, they look very good, and especially the surgeon, he's quite happy. So the same company then developed also heart valves, which can be resorbable, bioabsorbable, and these are the heart valves you can see here, which have been done and implanted in the Budapest, in Krakow, but also now I think also in Malaysia, in Kuala Lumpur. So 10 children were treated for that, and here about 16 children were treated with these heart valves. So you see, understanding, doing basic research leads usually at some stage to very interesting application, without which would not have been possible without the initial discoveries which can be made in basic laboratories. And it's considered as a breakthrough in surgical practice. Another aspect of supramolecular polymers is because the interactions between the components, the molecular components, are labile, that non-covalent, you can make films. This is a transparent film of a supramolecular polymer film, which can be cut in two pieces by scissors. These are the fingers of my Indian postdocs, Dr. Nabarun Roy. And you cut it, you superimpose the two ends, you push with your finger for a few minutes, and you can stretch it and it sticks. It's self-healing, and that's of course a property which non-dynamic polymers, non-supramolecular polymers, non-dynamic ones cannot have, don't have. Now, this being said, let's go back to the theme, which is part of it, step which we want to have to go to sort of end with. We have one other step to go, which is once you understand better how pre-organization functions and how molecular recognition can be performed through pre-organizing molecular matter through supramolecular interactions, then the question is, can we perform molecular recognition-directed self-organization? Which means we would like to generate complicated functional architectures by spontaneous way, in a spontaneous way, controlled by information and through recognition patterns, molecular recognition directed. This happens in this entity, which is the borderline between life and non-life, a virus. We know there's a virus running around right now, which puzzles us a lot. But viruses, as you know, maybe not all the students, do not live. A virus is a dead entity. And here I show one, the first one which has been well understood, the tobacco mosaic virus. It is self-organized, self-generated from its building blocks. 2,130 protein subunits represented here on the right. These are the protein subunits here, and you see a better sort of an amplification, an extension of this picture to have more details about what is there. What is apparent is you see that they bind together by their surface-to-surface interaction. And if you look at the left-hand side, we have this piece of cake, which, if it is well designed, gets together with another piece of cake, like here on the right, and then generates this circle, and then it can develop to generating sort of a tower, which has a hole in the middle. In the middle is the genome of the virus, and this happens spontaneously. Now, this looks like magic, but it is not magic. It is just chemistry, organic chemistry, structural chemistry, physical chemistry, and you can say analytical chemistry, all areas of chemistry together, and of course supramolecular chemistry, because the binding together of these basic molecules of proteins is a supramolecular binding. So the whole process of forming the virus is a supramolecular process. And this, of course, looks like a program system. I have insisted earlier on the fact that molecular information is stored in molecules and processed by interactions with molecules. That's exactly what happens here. The information is in the bricks, and the processing, the binding together, the way it happens is then determined by the way these bricks interact, by their shape and the way they interact. So this program chemical systems, the program is molecular, information is stored in the components, and the operation is supramolecular via recognition processes, which can be called an algorithm, the type of patterns which are used to process this information and to recognize one another. Let me just give you just simple examples of a category which is simple, not necessarily so simple, but a category of supramolecular architectures which is relying on molecules, organic molecules, which can bind metal ions. The bricks are the ligand molecules, and the cementing, the connectors, are metal ions. They bind to each other in a given way, determined by the molecules and by the type of metal ions, and then they can generate automatically, but in a very controlled way, given architectures. And a very great variety of that, such metallo-supramolecular, as they are called, architectures have been devised and have been generated. Here, this is the first ones we made, an artificial double helix, an artificial triple helix, made by wrapping 2 strands or 3 strands around, in this case, 3 metal ions. Here you don't see them on the left, but here you can see this yellow thing here, which are nickel ions in this case. These are crystal structures, not just nice pictures. Then you can also make grid-like molecules, molecules, grid-like, I should say, metallo-supramolecules. They are this specific grid is made from 3 linear molecules which are held horizontal, 3 other linear molecules vertical, and they are linked together at each intersection by metal ions. Or we can also say that the metal ions bring them together and sit at each intersection and provide this grid, spontaneously, of course. Here is an even more complex one with 2 different types of molecules. Red ones are linear, the blue ones are flat. They have points of interaction which are connected, which are brought together by connection with copper ions, copper 1 ions in this case. And this, just to illustrate this, there are here 3 linear, 4, this is 12 metal ions, 19. So these 19 components, when you mix them, they automatically generate this, what you may call a nanocylinder. Then there are many others. I show you here some because they look nice and okay, they're nice shapes, but many, many more have been made, much more complicated ones than those. And a number of those type of systems of type of architectures have also been used in this development of molecular machines. Now, what does that tell us? It tells us that we can generate complex architectures by handling in the correct way molecular information stored in molecules and then connect them together by, in some way here in terms of midlines. This is of interest also in nanoscience and nanotechnology, because we could say that this programmed self-organization can allow the generation of complex architectures, functional ones, through spontaneous assembly, to this self-organization of the entity. And this could be maybe not an alternative, but certainly a complement to nanofabrication and nanomanipulation, because it would represent the ultimate fabrication. Let the object make itself from the bricks. That's what a virus does. The assembly of a virus is a spontaneous fabrication of a supramolecular entity, the virus. And this is, of course, the ultimate, if I want to say, because it just happens by itself. But of course, you have to understand how to make it happen. So now I'm going towards the end, but I have to just point out something else which is of importance. What I showed you up to now is in supramolecular, on the supramolecular plan level, is obtained by design, storing information in molecule, programming these systems and let them go. And this then leads to the generation in a spontaneous way, but in a controlled way, of functional molecular and supramolecular entities. But there's another way. The next step is to rely on selection. In other words, not just have to design things, which of course is fine, but also see if we can set up systems where the formation of the organized architecture will rely on the selection of the correct building blocks from a diversity, a mixture, a set of building blocks. In order to do that, one also needs to have a system which is dynamic. That means that which can search the possible combinations. When two such entities get together, they shouldn't stick together too strongly, because then they may be sticking together in two entities, components which are not the best ones, the ideal ones, would stick together. And we have to have the possibility to search out the energy structure hypersurface. This then leads to the notion of a chemistry which is dynamic in its constitution. I just explain a little bit this because that's now mainly our work. A chemistry where the chemical object can fall apart and reconstitute itself. This can be done on the supramolecular level as well as on the covalent molecular level by introducing into molecules reversible chemical bonds. Now, since the dissociation and reassociation of components can incorporate or decorporate different components and lead to another final compound, it can adapt to what is going on, what is in the outside, in the world, which has an effect, an agent in the outside world, and there it leads to what one may call an adaptive chemistry. A chemistry of chemical objects which respond to what happens in the environment. Objects which will change and with the environment or with the application of a given agent, which can be simply protons or also going from a water medium to an organic medium and so on. I have no time to get into that. That is by itself several hours of lecturing, but just I want to make you aware of that. And let me just show you what it has already been used for. This constitutional dynamic chemistry, I insist again, either on the molecular or the supramolecular level, in the supramolecular level, it is inbuilt. It's built into the system because labile, the interactions are labile. In the molecular level, and on that level, one has to introduce reversible covalent bonds into the molecule so that they can dissociate into components and reassociate. This allows for adaptation through constitutional variation, and it can be used for searching for biologically active substances. That's been done for trying to devise ways of getting enzyme inhibitors, for instance, or substrates for receptors. The self-organization of nanostructures, which are dynamic, of course, and also the development of dynamic materials. Of course, I cannot cover all that, but it's just something when I come back to Crete, I will tell you about those things. So at this stage, and this is opening my conclusions, a few. From molecular chemistry, at the basis, we have to know how to make molecules. This is the art and science of molecular chemistry. But then these molecules can associate, and we have to learn how they can associate and how we can handle this potential of association and how we can direct the associations in a given way. They get organized because these interactions are specific. Dynamic is necessary for more complexity because you want the entities to be able to dissociate into components and reassociate in the same way or differently. Adaptive, they can, as I said, reassociate in a different fashion, adapting to the circumstances. And this is more and more progress and steps towards living matter. Now, I suppose that I have shown you that the essence of chemistry is to study progressively more complex states of matter, novel expressions, and even to create some which do not exist. In other words, the book of chemistry has to be not only read. Of course, we have to read the book of chemistry around us, not just living organisms. These molecules exist. But you can also make many, many molecules which don't exist yet. And so the book has to be written, or the score of chemistry has to be composed, not just to be played. So chemistry has basic creative power. And I want to illustrate that by taking a correlation, a relationship with art, which is considered as the human activity, the creative human activity. But science, not only art, science is creative as well. And we can illustrate that by coming back to Auguste Rodin, like the thinker on my first slide with the Big Bang. Auguste Rodin is one of my favorite sculptures. And of course, I don't want to speak about the very, very famous antique Greek sculptures in antiquity, which are world famous, of course, for many years. But I like this one because what is shown here, and what the Greek sculptors also practiced, to take a stone and make something out of that stone, which was not in the stone. And here the hand of the sculptor just expresses the organism of human beings out of the stone. Like Phidias was producing his sculptures from a stone, and a stone doesn't contain it. So chemistry does the same with the stones of matter, with the elements of matter. So chemistry can be called the art of matter. Now, this possibility of being very creative, for science to be very creative, was already sensed many, many years ago. Let's say this person, Leonardo da Vinci, who is very well known as a painter of La Gioconda, and but he was also a scientist, and he was an engineer. In 2019, it was his 500th anniversary, where it was celebrated all over the world. But what I'm interested here to show you is a sentence he wrote, which is a very, very strong sentence, which sort of covers what we have talked about. Where nature finishes to produce its own species. We are part of nature. We have been produced by the evolution of nature in the course of the evolution of the universe. Man begins using natural things. What is this? The periodic table of the elements, the basic bricks of visible matter, in harmony with its very nature. What is this? These are the laws of physics. What you can do, what is allowed to be done, and what is not allowed, what cannot happen, because the laws of physics tell you what is possible in visible matter to do. But then the ending is very, very strong, especially for an artist, to create an infinity of species. When you try to interpret that in modern terms, it comes back to what I said earlier. This is also genetic modifications to create an infinity of species. Species, not just non-living, but living ones. And I think Leonardo was right. Now, there's another one, another Greek fellow. You know, though, this one too. Prometheus, he stole the fire of knowledge from the gods, and he gave it to mankind. What a thing to do. Here he runs away, looking over his shoulder. He has the fire in his right hand here, and he sort of say, oh, the other guys are going to run after me and try to catch me. But they didn't catch him. And here he is, showing high, the brandishing in his hand, showing the light of knowledge. However, we have to know one thing. This is the fire of knowledge. That is what science gives us, but we cannot give it back. What we know, we know. We cannot erase it. What you know, you know. You can erase it if you blow up the Earth. That's a possibility. But some other planet will have also organisms which have knowledge and which are able to find out. They will also find the periodic table of the elements, of course, because there's nothing else. So we cannot give it back. This knowledge we have acquired, we have to live with it, and we have to try to take the best, to make the best out of it. That is not very easy, but we cannot do anything else. Our path leads us from the quest of knowledge ultimately to the control of our destiny. The destiny is now in our own hands, not in the spontaneous evolution of the universe, but in what we will do with it. And I'm sure we will use this possibility. The question is how? And that is the very, very difficult thing. Let me close here by citing also some mathematics, because I have not said anything about mathematics except some 1, 2, 3, 4, 5, which is very, very basic mathematics, of course, as you know. But here I show you David Hilbert, a very famous German mathematician. He's buried in Göttingen, and on his tombstone, he wanted to have engraved 2 sentences, which in fact he pronounced at a meeting. The sentences shown here are the following. Top sentence. Wir müssen wissen. We must know. This is already a sort of a strong conviction. Let me just sorry for that. The phone was ringing. So we must know. Wir müssen wissen. This is already strong. This is a goal. This is the strong expression of the goal of science. But then the second sentence, wir werden wissen, we will know. That is what drives us. The goal is to know. And the fact that we are convinced that we will know more and more about what we are doing here, what matter is, how the evolution of the universe occurred, and so on, and so on, and so on. This is really what drives us scientists, not just chemists, all scientists. Here again, Prometheus showing the light of science, brandishing, showing up high science, we will know. And especially for the young students, let me say, science shapes the future of humanity. Participate. Thank you very much. Yes, it has been a bit long, but I suppose, you know, I get carried away a little bit, so.
It was very inspiring. I mean, sometimes today we are often forget the history of chemistry, how important were the discoveries back then. And today we think that it's nothing and just surpassing. And of course, you really show that the limits of supramolecular chemistry are infinite. Let's start for some former colleagues or students of yours, which are now professors here in Greece, who I'm thinking that probably have something to say or comment. For example, my old professors like Professor Koubis or Professor Philaktakidou, maybe they want to...
I would like to say something. Although Professor Lehn... Hi, Professor Lehn. It's a great pleasure to hear this lecture. It's always inspiring. And I think all of us are your students, and we are very happy to listen to all these wonderful things. I would like to share with the rest of the audience that when I was in Strasbourg like 20 years ago, it was in 2000 to 2001.
It was 20 years ago. It's incredible.
Yes, it is incredible. I want to share with the audience that we had these group meetings on Thursday evening every week, and we had a time to start, but there was no ending. And I remember sometimes your wife was calling you to go for dinner, but we had our group meetings with beers, with snacks, with everything. And I still remember all this power you have when you talk or when you share things with your students. And I wish you the best to continue all these things you are doing. I'm very touched with all the things I saw here. So I don't want to say anything else, but I like this slide with the Garden of Eden, with Eve and Adam.
Oh yes, I should have done that.
with the apple. I prefer the apple, that we won't give back this apple of knowledge. Thank you very much.
Thank you, Tina. Very nice. You know, it's difficult to realize that it was 20 years ago.
Yes. Yeah.
And thank you, you and Alex, for what you have done, because ITPP, I will see, this compound is still now in the clinic, and there are some positive effects. But of course, for the moment, the phase 2 is still a bit early. So we will see what happens. And it's being continued by the people in Zurich.
Okay, thank you.
Professor Pizzinos.
This is Mike Karabatsos.
Yeah? Oh, how are you?
I'm fine. I've enjoyed your lecture tremendously. Yes.
Where are you right now? Where are you?
I am in the United States. I'm in East Lansing, Michigan. I'm an honorary professor also at Michigan State University. I have my office. I still go. I'm a little bit older than you are. I was born in 32 and you were in 39. Well, what, 7 years difference? So, yeah. But I still go to my office. And,
yes.
I learned many things. I've enjoyed it, and I hope we'll get the chance to meet in Crete again.
Oh, yes, it would be a very great pleasure, you know, that's for sure. And, you know Jim Dye also from Michigan, Michigan State University in East Lansing. He is also has been working a long time. He was in the 90s. Now he's a bit not in so good shape. But this is the one, you know, the person who made sodium minus, potassium minus, and things like that. Anyway,
well,
it's very nice to hear you. And thank you very much for joining in. Of course, this lecture was for students, but okay.
There are a lot of people that want to talk to you. And Professor Pizzinos, you may want something to add.
Oh, Where is Mike? Where is he? I don't see him.
There are so many people attending that it's
Here he
difficult to
is.
visualize
How are you?
everybody. Professor, my congratulations. The same thing that Dina mentioned. It's always a pleasure listening to you, presenting science, because you give the larger picture. Sometimes we get too much absorbed by our particular problems that it's useful to be reminded what the big question is and what we have to be going after. So without taking time from perhaps other people that want to comment, thank you very much again for a wonderful lecture. And of course, for the opportunity you gave me many, many years ago, 20 plus, to work with you and be motivated by your way of thinking.
Thank you very
much. All the best.
Thank you. I just noted that I have added another slide, and this I would like to show for the students, because I would like to comment on it just briefly. Allow me that. Science has another aspect which one should really make students understand. Science is worldwide and even, I would say, universe-wide valid. It has no borders of nationality, no borders of flags, no borders of dogma, of religion, of whatever things separates people. And it is the same everywhere. Science has no borders. And it means for us human beings on our planet, then when we go around the world, you have friends, people you know everywhere. You know, when you go around, you are in a city, and if it happens that there's a chemistry department, you can go there and say, okay, I am whatever I am, and we can talk science. And I think this is possible, really. And this is worldwide, and as I said, also universe-wide. If someday some of the people on the planet Earth can meet people or some organisms on the other planet, organisms who have also then devised enough knowledge and so on to build up science, they can talk together, and probably they will even have automatic translation at some stage. Okay, this is just to add something, because students realize that.
That's very, very important. Maybe Professor Zivgoulis, you want to add something, or you are muted. Okay.
Yes, greetings from Patras. How are you, Professor?
Oh, yeah, Makis, how are you?
Congratulations, first of all, for this excellent event. And congratulations, Professor Lehn, for this inspired lecture. Always you give amazing lectures. What a nice start for this new year, really. I would like, since your lectures always have a philosophical dimension, since I know you 25 years ago, I would like to make a question, if it's allowed, if you allow me. So let's say that one day we will discover life in another planet, which I believe it will happen sooner or later, and I know that you believe it too. Will be based on carbon. What is your opinion? You mentioned something about the DNA molecules, but I will go one step backwards. So it will be based on carbon. Thank you again for your nice lecture and your presence here.
You see, we have not worked on that, but I think the conditions and the properties of the elements are such that carbon offers the best possibilities. It could be on a very hot planet that silicon might be part of the game, because silicon-oxygen bonds and so on, you know, strong bonds and so on. Or if it's a cold planet, it could be weaker interactions, but it's almost inescapable that it should be carbon. It's not, you cannot say it will be, but it's a very good chance. And there is a book by Christian de Duve, who got a Nobel Prize in Physiology Medicine. Its book is Vital Dust, the title of the book, Vital Dust. And the subtitle is Life as a Cosmic Imperative. I have just extended it, not just to life, but everything is an imperative, a cosmic imperative. And life is part of it. So I'm quite convinced that life exists. I also think that the conditions, if we, especially, let me go back, if we look for planets where the conditions are similar to Earth, then it is almost certain that it will be based on carbon. Because you have to make bonds, you have to break bonds, you have to rearrange things, and how can you do it? It's if the conditions are the same, then the same type of, the constructions can be different. The letters for the genetic code or whatever, or they can transmit maybe information in a totally different way. But there will be, the organisms will probably be based on carbon.
Thank you, Professor Lehn. If I may ask another question too.
Go ahead.
So what do you believe, Professor Lehn, will be the future of supramolecular chemistry? Is it possible that in the near future, let's say we will be able to mimic what cells do, the cell actions, so do reactions with mixtures, stable mixtures, and each molecule finds its partner to do the reaction?
So the question would be?
The question is what it will be the future of supramolecular chemistry. What is your opinion?
Oh, you know, it is like if you had asked the same thing 100 years ago to chemists, what is the future of molecular chemistry? Huh? I think it's the same. It's a field which, look, I mean, molecular chemistry was around also when Democritus existed. Supramolecular chemistry was there when Democritus existed. Huh? But okay, then the fields develop, and once they are set up, they continue to develop. I think molecular chemistry still has a very bright future, and we see every year, every day almost, new products, new ways of handling things to think about. Supramolecular chemistry is now an established field. It's about 50 years old, and it will continue, and we will exploit other features. Maybe going closer and closer to the one goal is quite obvious, that living systems have a very complex organization. So the fact that living systems, that life and thought exist, look, I said already, it's a tautology. Huh? The fact that thinking exists proves that it is possible. I don't say much by saying that. It's not a big discovery to say that, but it means just that it is possible. And that encourages us to try to understand how it is. And once we understand how it came about, I cannot predict what happens. That would be science fiction. But very probably we'll be able to understand what has happened. Possible even that we may go beyond it. But then it's the ground gets very, very, very dangerous, very difficult to know what to do, but we will be able to know it, and we will be able to progress more and more. And the future of supramolecular chemistry is exploiting what it has brought and going further, going beyond.
Thank you, Professor Lehn.
There's a lot to do still. There's in applications and lots of things. Look, let me just give an example, because that's very present day. You know, now we have this virus, which is making havoc out everywhere. That's why I'm not in Crete right now. This virus is not even living. But we have now vaccines coming, the messenger RNA vaccines. Now, the messenger RNA vaccines were developed, and that's also an interesting story about how things can happen and how things can occur in a way which is impossible to predict. A lady scientist from Hungary, Katalin Tarico. She came from a very poor family, and she immigrated, and later on, I don't have the dates like with me, but she immigrated to the US. She did her studies there at Temple University, and then was hired by University of Pennsylvania, UPenn. She was convinced that messenger RNA is very, very important in vaccination and in many, many other therapeutic aspects. But of course, messenger RNA is very sensitive to degradation. And also, it is recognized by the immune system and then leads to very strong inflammation. But they found out, then she went together with a colleague. No, first of all, I should say, that's also something for you, that she was not recognized by the scientific system because people said it cannot work. She got no money, all her grants were turned down. But she insisted, she hung on. She was very tenacious. She got together then with a biochemist, biologist at UPenn, Drew Weissman, and together they developed the basis of what is now this vaccine, which is the best. I mentioned that also for another reason. The basis is chemistry, because what did they do? They looked whether or not one could exchange the nuclear bases in the RNA, so it would not be recognized by the immune system and not lead to these adverse effects, and also become more stable. They replaced uridine by pseudo-uridine. That's a very simple chemical change. And the present-day vaccines are based on that. The messenger RNA with pseudo-uridine are much more stable. They do not elicit the immune response, and they are much more active in giving rise to immunity. So this is a story which is incredible. Somebody from a small village, small town, poor town, very poor family, going there, hanging on, and here it is. I should also say to that there's something else, and I think for you Greek people, it is sort of close to you, but the company which then developed it, BioNTech in Germany, was set up by two Turkish medical people who went to Germany and developed it. They also had maybe no other, they were even clandestine. What is that again? The father of these two, the father of the man, it's a couple, a lady and a man, and her husband. He was a clandestine, worker, a worker who was not allowed. I mean, not an open. Yeah, he went into Germany, and then they developed it. So you don't know where science is going to happen. And it's incredible.
Professor Lehn, we have a time limit, unfortunately, with Zoom. So that's all very inspiring that we can talk for hours. But let's take one more question that George Petekidis wrote in the chat. It says, thank you for the inspiring talk on the links of chemistry life. It seems to me that a fundamental notion, phenomenon in this link that you did not touch upon today is the role of entropy and Brownian motion. This, together with interactions, is behind what you called magic in the self-assembly in all mesoscopic, soft, and biological systems. Could you please give us your view on this?
Yeah, of course. This is a question, which one can ask sort of first sight at. What about entropy? What I told you today is, of course, simple thermodynamics, and it's not even out of equilibrium. And we know that life if we want to be, yeah, the question before, which was asked about what's the future of supramolecular chemistry. I think one very important future is, I come back to your question in a moment. One very important question is the setting up out of equilibrium system, having energy dissipative systems like a living system. Living system is of course unstable, but it runs, and it runs because it is an out of equilibrium system which can feed itself and so on. Now, this is one of these fields which is being developed and will develop more and more. Now, coming back to the question of entropy. Entropy is totally, will always, of course, we know the second law of thermodynamics and all that. We cannot go, we cannot go against that. I mean, nothing is going against it. But what is true that when order can be generated, and of course there is disorder somewhere else. And for instance, let me just a very simple example. When you have two hydrogen atoms, they have 3 + 3 degrees of translation, 6 degrees of translation, 3 for each particle, each atom. You make a hydrogen molecule out of it. This molecule has only 3, so they have lost 3 degrees of translation. How is it possible? Because it's the enthalpy, the energy released by making the bond. So it's delta G which rules the world. And when there is a loss of entropy, then it is due to the fact that you have enthalpy which compensates for that.
Okay, thank you.
Thank you, Professor Jean-Marie Lehn. It was a great pleasure, a great seminar, very inspiring. And but unfortunately, we exceeded the 2 hours and the 500 participants. So
500. Oh, that's nice. This is one of the biggest number I had. No, yeah. True. I've been giving Zoom lectures. You know, nowadays I am traveling around from one country to another one by Zoom. And 500. That is, I think that's close to the record. Maybe in China have a little bit more, but Greece is much smaller than China.
So I would like to thank you and thank everybody for participating in this very special seminar. And Jean-Marie, you are very welcome whenever you like to visit your department, the Department of Chemistry, University of Crete. Thank you all very much.
Yes, but if I come to Greece, I will come, of course, to Crete, but not only.
Definitely.
Yes, Patras too.
We get friends around. I mean, you see, it's...
All right. Thanks for coming around. Thanks for the invitation. It was very nice to see some of my former collaborators. It's fantastic to see them. Tina, hello, hello. Where is Alex? He's somewhere around. I don't know where.
Hello, Professor Lehn. How are you?
Oh, here is Alex.
I'm sorry, my camera has a problem. How are you? It was really very nice to listen to your talk again.
Okay, okay. So carry on. Huh? Yeah, as I said, 2,500 years ago, you had top scientists. Continue.
Okay, thank you.
Okay, bye-bye.
Thank you all very much.
Bye, everybody.
Thank the organizers too.
Enjoy the weekend. Huh? Okay.
Thanks. Bye-bye.